Hey guys, so this video is going to be over regulation of the cell cycle. I'm going to try to handwrite this stuff because I'm getting a little bit better at it, but if not, then I will start typing stuff out. So here we go. So the regulation of the cell cycle is really important because it essentially is how you heal yourself. When things get out of whack, that's when things like cancer and growths happen. Okay, so understanding how the cell cycle is regulated is really, really important for many, many things. And it's the subject of many research labs all across the country. So it's a really important thing to do. So let's Okay, so if you remember from my last video when I was talking about cell transport, I made a comment about how cells essentially need to be a certain size. So they can't be too small because you've got the cytoplasm and all the organelles that need to fit into it. And it can't be too big because if a cell is too big, then it's not going to be very efficient at getting things out of the cell that needs to get out and things are getting things like nutrients and other substances into the cell that it needs. Okay, so it needs to be a certain amount of size. That's the good surface area to volume ratio that a cell likes to kind of keep intact. So you have to keep that in mind. We're talking about cell division because a cell will not divide if it's not big enough. Okay, so there's a couple things that will determine when a cell divides. Okay, so one of them, it's got enough cytoplasm. Okay, so one of the things it has to have enough of is the organelles, but also the cytoplasm because one cell will at some point, more often than not, will divide from one cell into two daughter cells. So the parent cell has to have enough cytoplasm to make two fairly good sized daughter cells so that they can grow from when they're made. Okay, so they got to have enough cytoplasm. Okay, the same token, the cell that is going to divide has to have DNA that's been replicated because those daughter cells need to have exact copies of your DNA. Okay. So the DNA, ooh, that is not a W. DNA is replicated. Right? In order to, again, nourish itself and sustain itself as cell division is occurring, okay, that cell has to also have enough nutrients. You know, just like you can't expect you or your kids to have a productive day when they're at work or at school, a cell can't be productive if it doesn't have enough nutrients. So there's no way it's going to be able to divide if it doesn't have the nutrients to sustain that division process. Okay, now kind of where it gets a little complicated is how that cell division is signaled. Okay, and that's what we're going to talk about today. So it has to be stimulated by what we call growth factors. These are chemicals that are going to signal the cell that it is time to start growing so that it can then replicate or divide and make those daughter cells. Okay, so it has to be stimulated by growth factors. And again, those chemicals can come from a bunch of different places. The kidneys is one place. It can come from other cells around it. If a cell gets damaged, it can give off hormones essentially or signals to the cells around it that it needs to start dividing so that your body can heal. Okay, so those are growth factors. And then the last one, number 5, the cells around it will die. Okay, so when the cells around a living cell die, what that does is it opens up space. So one of the things that regulates how much a cell will divide or how quickly cells will divide is whether it's touching another cell or not. Okay, and there's this thing called contact inhibition. Okay, this contact inhibition is what turns off the division process when the cells are sufficiently packed in to, again, maintain that barrier between inside and outside, especially on your epithelial cells, the skin. Okay, so that contact inhibition essentially means that growth is inhibited. It stops as soon as it touches another cell around it. Okay, so when you give yourself a paper cut, your body knows that's healed when the new cells butt up against new cells from the other side of that cut. Okay, so that makes it stop. Without that contact inhibition, we get uncontrolled growth, we get cancer, that kind of stuff. So contact inhibition is pretty important. All right, let me go to the next slide here. All right, now, what we do know is that the cell cycle is regulated at checkpoints. Okay, and hopefully what you know from reading a little bit earlier in, I think it's 4.3 also, you talk about the cell cycle itself. Okay, so the cell cycle includes interphase, prophase, metaphase, anaphase, and telophase. Okay, if you can remember IPMAT or PMAT, anything like that, that's going to help you to remember those in order. And in all honesty, as I was writing those down, I was going through this set of letters to make sure that I was putting them in order. So IPMAT, and then at the end of that you have cytokinesis. Okay, and most of the time cytokinesis and telophase kind of happen at the same time. So that's why we don't have a C in this abbreviation here. And if you wanted to put that in there, what I learned, it was my first year of teaching, I had never heard a sentence to go along with this. I always learned IPMAT, but the teacher that taught next to me, when he taught this class, he would teach his students the phrase, something along the lines of, ingenious pimps make all the cash. And he told me that, and it was like a light bulb went off. I was like, wow, that's a really interesting but good way to remember that. So if you can remember that sentence, those should stay in order for you. Now, I'm going to try to draw this out. I'm not the best drawer, that's why I'm not an art teacher. So if I draw out essentially what the cell cycle is going to be, okay, you'll see this figure in your book on page 127. Okay, so most of the life of a cell is going to be spent in interphase. Okay, but interphase itself is divided into three sections. Okay, so all of these here, that kind of putting a line of blue in here, and again, I'm sorry for my kindergarten-esque drawings. Okay, all of those are interphase. Okay, this part here, I'm going to do in red, is what we consider collectively is mitosis, or the mitotic stages. Okay, those mitotic stages are these, oops, I'm sorry, got a little bit out of my own way here. Those mitotic stages, let's try this again, are prophase, metaphase, anaphase, telophase, and cytokinesis. That's what fits in this red area here. Okay, so I'm not going to try and draw those out because it's I don't trust my my drawing skills on this pad yet. So interphase, as I said, has three stages. Okay, so you have G1. Okay, G1 is the first growth phase. You have S, which is your synthesis phase, and you have G2, which is your second growth phase. Okay, and within this whole cell cycle together. So the cell cycle is all 6 of these: interphase, prophase, metaphase, anaphase, telophase, cytokinesis. Mitosis itself is just what I've put here in red. Okay, there are checkpoints throughout this that your cell goes through to kind of make sure things are going okay. And then if things are not going okay, then, excuse me, then your cell will actually stop what it's doing and make corrections, or sometimes it will just stay in this indefinite stage of being. Okay, so your first stage here, first checkpoint, is going to be here between G1 and S phase of interphase. Okay, this is what we call, well, it's this, essentially the starting checkpoint, if you will. But this is one where make sure that after cell division and that the daughter cell has been made, that everything's going okay. So there's enough cytoplasm that's being made. There's enough organelles that have been made to kind of sustain the cell. Okay, if there is not enough, okay, the cell will go into what we call a G0. Okay, G0 is kind of this indefinite cell life. Okay, it's not going to divide, it just kind of does its thing the whole time. So that's only if the cell does not pass G0 here. Okay, if it does, it will then go into the synthesis phase where the DNA is going to be replicated. Okay, and then it will go into G2. So G2 is essentially the cell getting ready to replicate. So a whole bunch of different organelles. We've already replicated our DNA. We're essentially just making enough things so that we have enough to divide between our two daughter cells. Okay, so the next checkpoint to make sure that all that has gone well is right here. Okay, this is the start of mitosis checkpoint. Okay, if the cell is not ready at this point, it will kind of stay in G2 until it is ready. Once it passes that point, then it will go into mitosis and will begin the division process. The last checkpoint is going to be between metaphase and anaphase. I'm going to put this over here because I didn't really write in this pie piece kind of where that checkpoint was, but I would say if I were to put your pie slices in here, it'd be right here. But that one is this guy right here. So it's between metaphase and anaphase. Just again, it's just making sure that all of, especially at this stage, at this third checkpoint, it's making sure all those sister chromatids have separated and there's not any kind of non-disjunction happening. So non-disjunction is the failure of those sister chromatids to separate. So we're making sure that everything's separated like it was supposed to and lined up in the middle and all that stuff, okay? So that's kind of the gist of the cell cycle. But with these checkpoints, it is regulated by two things, okay? Two essentially proteins. So one of them is called cyclin, okay? And the other one is what we call a cyclin-dependent kinase. Okay, so as the name suggests, these cyclin-dependent kinases, or you see them as CDKs, these CDKs are dependent on the presence of cyclin, okay? There's a whole bunch of CDKs that are always in the cell, but essentially what regulates the cell cycle and tells the cell when to start going is the cyclin here. This cyclin will fluctuate depending on what part of the cell cycle the cell is in. Okay, if it's getting close to the divisions part, there'll be a lot more cyclins in that cell. Okay, those are, and essentially what happens is those cyclins again will bind to the CDKs, and that will start a cascade of reactions in the cell, which will get that division going. Okay, this cyclin, when everything is done, okay, is broken down by molecules that we call proteasomes. Right, and this, the breakdown process of cyclin, is what turns the cell cycle off when everything is done. Okay, but what happens is if there's an issue with these proteasomes, and of course other things, it will not turn off those cyclins, and the cell cycle will continue and will keep dividing, and that's when you get an overdivision of cells. You get too many cells in one area, and then you get growth and cancer and things like that. Okay. Do you have any questions about cell cycle checkpoints, please let me know. But that is all for this.